thank you for this opportunity to present my research. I'm really sorry that the title in the book is not the same with the title that I proposed to this seminar. My research is a genotype I and IV of dengue virus, DEN1, and its clinical manifestation in Surabaya, Indonesia in 2012. This is the work of our colleagues between Universitas Airlangga and Eijkman Institute of Molecular Biology in Jakarta. We will perform the research in Surabaya, which is in this area, just as Professor Aryati explained earlier. And as we know, dengue virus is a genus Flavivirus with family Flaviviridae with genome 10,700 base. This virus is a single-stranded positive sense RNA. So it consists of 4 serotypes, which is DEN1 until DEN4. This is the genome structure of dengue viral. There are part of structural genome and non-structural genome, and it's forward from 5-axis untranslated region to 3-axis untranslated region. The gene consists of structural is C, PRM, and envelope gene, and for non-structural genome, it consists of NS1, NS2A, NS2B, NS3, 4A, 4B, and 5. As a background of our research is we know that DHF or dengue hemorrhagic fever incidence in our country was still number 2 in Southeast Asia, and the mortality rate is still over 1%. Even we increasing the treatment over there, and pathogenesis is still controversial. There are many theories proposed by several researchers, and most scientists agree with antibody-dependent enhancement theory proposed by Halstead. This is the antibody-dependent enhancement theory. When the dengue viral infect by mosquito bite, the dengue viral will attach the cell, which is specifically majority is dendritic cell, and the cell will engulf the viral, and the viral will replicate within the cells. After it mature, the cell will burst and expel the dengue viral with many more from the earlier. And after that, there are activation of T cell and B cell. B cell will produce antibody that can neutralize the viral. So when it is neutralized, it is inactivated and can destroyed by macrophages. But when somebody gets second infection with other type of serotype of dengue viral, the memory cell already there and could rapidly produce antibody. With this antibody is not homologous, but it is heterologous and cannot neutralize completely of the viral. So it can serve also as a entry site to the macrophages to Fc gamma receptor, and will promote more dengue replication, and will have a severe manifestation. So while there are agreement to ADE theory, but it has controversy. Why? Because there are proof that there are occurrence of DHF and DSS in primary infection, why it is still occur. Well, we know that on secondary infection, the manifestation is more severe than the primary, and there is a low case of DHF while the area was underwent secondary dengue infection epidemic. So we think about another factor that contribute to clinical manifestation, and one of the factor is maybe a viral virulence factor. So viral virulence factor already known with dengue 2 viral. In Southeast Asia genotype gave severe manifestation than American genotype, and they found molecular determinant as protein A, amino acid 390, and other gene such as 5-axinin translated region, PRM, NS1, NS3, NS4B, and NS5. But the limitation of dengue viral 1 exploration still exists. So we want to explore dengue viral 1 because in our region in Surabaya, there are safe from other serotype to dengue 1 serotype, which is in 2005, Professor Ariati work conclude that there are no dengue viral serotype 1, and the majority is dengue 2, followed by dengue 3 and dengue 4. But our study in 2012, we found that dengue viral already persists there, but majority is still dengue 3 as 47.4%. But in 2012, our research, another researcher, but in our region also found that dengue 1 serve a majority of the dengue viral infection there and consists of genotype 1. So objective of the study is to analyze the relationship of dengue viral 1 genotype and its clinical manifestation. So the methods of this study is observational cohort study with subject adults and children. We did a routine laboratory examination and molecular laboratory examination. So 101 subjects were children under 14 years old and 47 were adults, and we performed in region of Surabaya. All patients underwent 2 until 4 times examination, complete blood count, aspartate aminotransferase, and alanine transaminase, and albumin. We want to know this parameter as a parameter to know the severity and the clinical manifestation based on WHO CRO 2011 guidelines. We perform anti-dengue IgG and IgM to detect whether it is primary or secondary infection. We serotype the samples using firstly by 2-step conventional RT-PCR, then we confirm by real-time PCR using Simplexa real-time PCR machine. And we cultured positive result in C636 cells and harvest in 14 days. And samples that gave culture positive result were genotype using envelope gene sequence. We sequence using dideoxyterminator in Eijkman Institute molecular biology, and genotyping classified by Goncalves criteria, and phylogenetic analysis is done using Mega version 5 software. This is the patient characteristic. The male is 70 and female is 78, no difference. And the age children among the majority of subject here, which is 101. And the diagnosis consists of undifferentiated fever, dengue fever, DHF, and expanded dengue syndrome. We got positive PCR 84 and negative 64. When some of dengue serotype, we found mix of dengue serotype, which is 1 and 2, 1 and 3, and 1 and 4. And majority of serotype is DEN1. On children and adults, majority of dengue viral infected patient is DEN1 and followed by DEN2, DEN3, and DEN4, and found mix also. On laboratory result of different serotype in children, we found no significantly different between DH or DF or DHF or day of hospitalization and viral load. The CBC also has not so significantly different, and also AST and ALT, no difference between DEN1, DEN2, DEN3, and DEN4 also. On adult, as the same of the children, no different of clinical manifestation. There is hospitalization and viral load, but there are MCHC different in adult, basophils, neutrophil, lymphocyte, and monocyte between serotypes. No difference between AST, ALT, albumin, and secondary manifestation. This is the phylogenetic tree of DEN1. I'm sorry it is so little image. So we found only genotype 1 and genotype 4 on our samples. Based on genotype, we classified in children and adults. Mainly, there were no different significantly, but in erythrocyte, MCH, and lymphocyte, it different significantly in genotype 1 and genotype 4, also for albumin. On adult, we found no significant difference. This is the amino acid sequence variation in dengue 1 samples, which is in several amino acid on 103, 104, 109, 155, 171, 2003, 203, I'm sorry, 227, 293, 338, 339, we found variation that changed the nature of the amino acid from hydrophobic to hydrophilic, or from hydrophilic to hydrophobic. So it could be changed the conformational structure of the envelope, but again, it not correlated. It didn't correlate with the severity. So conclusion is there were no significant difference between clinical manifestation of DEN1 genotype 1 and 4, and from gene, amount of amino acid substitution had no implication in DF and DHF manifestation. Thank you.